Hello everyone, I'm Anas Al Ubaidi, and this is session 25 or lecture 25 within chapter 24 of the Advanced Medical Science Liaison and Medical Monitor Certification course program by CCRPS. The chapter is about conceptual and theoretical monitoring, while the topic for the session is about monitoring of drug accreditation and drug certification in drugs and trials for physician medical monitors or for for medical monitors. Introduction. Definition. Drug accreditation and drug certification in drugs and trials focus on validating drug safety, efficacy, and quality through established accreditation and certification standards. These trials ensure drugs meet international or domestic regulatory benchmarks required for approval and market entry. Key points. Accreditation assessment definition. Accreditation assessments evaluate the extent to which a trial facilitates data management systems and protocols adhere to guidelines from accrediting bodies like WHO, World Health Organization, FDA, Food and Drug Administration Agency, or EMA, European Medicines Agency. And here's a summarized diagram summarizing the accreditation process for trial readiness from top to bottom: trial facilities and protocols, good manufacturing practices, good clinical practices, technical validation, compliance challenges, and their successful accreditation. The focus area in accreditation assessment, we have good manufacturing practices, GMP. GMP stands for good manufacturing practices as one of the focus areas. Ensure that production, GMP, good manufacturing practices, ensure that production facilities meet required technical and operational standards. As for example, a vaccine trial ensured GMP, good manufacturing practices, compliance by maintaining sterility and by containment during live virus handling or live virus handling. Good clinical practices, GCP, verify that clinical trial processes meet ethical standards such as patient safety and informed consent. As for example, a phase 3 oncology study reviewed site accreditation to confirm adequate patient data management aligned with GCP, good clinical practice. Technical validation of facilities include facility inspections and equipment audits to establish readiness for trials. Challenges. Complexity of maintaining continuous compliance across multi-center global trials. Another challenge, meeting accreditation requirements in regions with weaker regulatory frameworks, another challenge. The outcome here, successful accreditation confirms the readiness and reliability of the clinical and manufacturing processes, ensuring a seamless drug development timeline. Certification evaluation. The definition, as for certification evaluation, focuses on evaluating specific drug parameters such as potency, stability, stability, and purity to ensure each produced batch meets regulatory standards. So the certification evaluation here, shortly or in simple, focuses on evaluating specific drug parameters. And these specific drug parameters, such as, or might include, not limited to, as for example, the most common ones, potency, stability, and purity, to ensure each produced batch meets regulatory standards. And here the key measures: batch to batch consistency, as regularly verify all batches meet predetermined specifications. Example, during an oncology trial for a biosimilar, active ingredients levels were certified by the EMA, European Medicines Agency, to ensure uniformity before prior to larger scale production. And here, biosimilar in European Union, particularly in EMA, through the EMA, means medications that having the same contents, chemical contents, same biochemical contents, the same effectiveness, and the indication for the original brand name medication. And in European Union, EMA, it's called reference medicine. Uh, the brand medication or the original medication called reference medicine or reference medication. And biosimilar in EMA means like how it is named by the FDA in USA generic, meaning the medication that's similar to the original one. Drug stability confirmed that drugs remain effective and safe within their shelf life under various storage conditions. As for example, a diabetes drug underwent stress testing to assess stability at different temperatures prior to before receiving certification. Potency and purity testing ensure that active ingredient concentration is maintained without contaminants or harmful additives. Example, certification of a rheumatoid arthritis, RA biologic involved chromatographic validation of its active monoclonal antibody. Challenges lengthy stability testing periods that may delay qualification or certification timelines. Another challenge, variability in batch production due to discrepancies in raw materials or environmental conditions. The outcome certification evaluation ensures the reliability and safety of drug batches, earning the confidence of regulators, healthcare providers, and patients. Data integrity. Definition. Data integrity refers to ensuring the transparency, accuracy, and reproducibility of data collected and reported during trials. Strategies, and before we start with that, we have here a diagram on ensuring trust and compliance with data integrity in trials. From left to right, we have audit trail systems, electronic data capture, data protection plans, all related to one another in order to reach the data integrity. The strategies here, audit trail systems implement comprehensive documentation processes validated through regular audits to align with regulatory audits. Example, a rare disease orphan drug trial used 21 CFR Part 11 compliance software to log stability data ensuring FDA readiness. Electronic data capture EDC centralized systems help track participant data, patch testing results, and quality metrics in real time. Example, an HIV therapy study recorded all pre-certification test results into an FDA-compliant platform, ensuring clarity and reproducibility. The data protection plan secures systems against unauthorized access or unintentional errors through encryption and version control. The challenge is here synchronizing data across diverse locations. Another challenge preventing discrepancies when transitioning between research, development, and certification phases. Outcome maintaining data integrity builds trust in trial results, ensuring regulatory approval and stakeholder confidence. Examples of successful trials: WHO, World Health Organization vaccine accreditation established compliance with sterility and cold chain protocols, enabling global distribution. Anti-diabetic batch certification ensured uniform efficacy in injectable insulin pens produced at multiple facilities. Multinational generic certification simultaneously aligned with European pharmacopoeia and USP standards. Rheumatology biosimilar trials, FDA biologic approval was streamlined through early phase accreditation integration. Pediatric antibiotic certification, real-time deviation management, flag formulation inconsistencies, ensuring patient safety. And here's a So diagram summarizing the diverse pathways to pharmaceutical compliance. As on the left, we have pharmaceutical certification and accreditation. On the right, we have the WHO vaccine accreditation, anti-diabetic batch certification, multinational generic certification, rheumatology biosimilar trials, and then pediatric antibiotics certification. Summary tables. Next, we have tables. We will have the tables that would summarize what we have covered through this lecture. As we have table on key differences between drug accreditation and drug certification interaction trials aspect, and for each aspect comparison between drug accreditation and drug certification interaction trials. As for the aspect of definition for drug accreditation interaction trial assesses the compliance with regulatory and ethical standards. As for drug certification interaction trial verifies drug specific quality like potency and stability. Other aspects to compare, like main focus, governing bodies, primary objective, and then the example. Actually, in governing bodies, for the drug accreditation interaction trial, we have WHO, FDA, EMA focusing on GMP and GCP, good manufacturing practices and good clinical practice. While for the governing bodies for the drug certification interaction, we have National Pharmacopoeias ISO 13485 for product evaluation. Table on key components of monitoring and accreditation and certification. Component focus example. Components we have accreditation assessment, certification evaluation, data integrity, batch consistency, and deviation management. Another table on tools and techniques for monitoring accreditation and certification. We have tool technique purpose example. Tools or techniques, we have quality management systems, QMS, compliance audit tools, digital dashboards, laboratory assays, and supply chain tracking. Table on steps for effective monitoring and accreditation certification. Step focus examples. Steps we have protocol development, real-time data collection, pre-certification audits, risk mitigation, and then reporting. Table on real-world applications of accreditation and certification monitoring, trial scenario, purpose outcome, trial scenarios. We have vaccine trials, anti-diabetic certifications, generic globally certified drugs, biologic rheumatology, a drug, and then pediatric antibiotics. Application table. For application table, we have scenarios. For each scenario, key principle applied, key actions, timeline, what to watch out for. Scenarios, we have ensuring facility compliance for vaccine cold chain integrity as part of WHO accreditation, verifying batch consistency for oncology medication to meet EMA certification standards, maintaining data accuracy in a rare disease drug trial for compliance with FDA mandatory documentation. Monitoring blood pressure effects in a cardiovascular drug under ISO 13485. Device certification protocols, real-time monitoring of tablet dissolution to ensure consistent quality in cardiovascular drug testing, addressing excipient sourcing violations in dermatological cream trials before prior to final certification. Validating HIV drug stability through cell-based laboratory assays during WHO approval trials, preparing biologic drugs for FDA inspection through simulation audits of manufacturing environments, tracking deviations in insulin pen production for anti-diabetic certification under GMP guidelines, performing surprise audits in oral contraceptive manufacturing to prevent contamination risks. Next, we will revise what we have covered through this lecture through some MCQs, multiple choice questions. Question 1, what is the primary purpose of drug accreditation interaction trials? A, to analyze individual patient responses to treatment regimens. B, to ensure compliance with the GMP and GCP standards for regulatory approval. C, to identify drug design flaws in preclinical stages, or D, to assess patient satisfaction with medication regimens. So definitely the answer is B, as to ensure compliance with GMP and GCP standards for regulatory approval. As drug accreditation interaction trials focus on ensuring that manufacturing and clinical practices align with regulatory expectations such as GMP and GCP, which are critical for drug approval. Question 2, what is the key focus of certification or evaluation in drug trials? A, establishing genetic markers for therapeutic responses. B, verifying drug purity, potency, and batch consistency. C, monitoring long-term efficacy in post-market scenarios. D, assessing trial participant adherence to protocols. So the answer is B. As for certification evaluation in drug trials, verifying drug purity, potency, and batch consistency. As certification evaluation ensures that the drug batches meet specific quality parameters like purity and potency, which are essential for regulatory certification and patient safety. Question 3, which tool is most effective for addressing deviations during monitoring? A, ECG monitoring devices. B, deviation management systems with kappa tracking. C, wearable sensors measuring patient vitals. Or D, routine paper-based batch record keeping. So it is about tracking or addressing deviations. So definitely the answer is B, as of deviation management systems with kappa CAPA tracking, as deviation management systems automate the detection, documentation, and resolution of issues, ensuring that corrective actions kappa are efficiently implemented. Question 4, what is the primary role of quality management systems, QMS, in drug trials? Is it A, to provide visual dashboards for patient-reported outcomes? B, to track and resolve deviations while enhancing compliance audits? C, to collect real-time patient blood samples during trials? Or D, to limit external audits for internationally distributed drugs? So it's key QMS, quality management systems, is to track and resolve any issues within quality, maintain quality or quality control. So definitely what applies between the four choices and between the four options would be B, to track and resolve deviations, as the less deviation, the better quality of the trial, while enhancing compliance audits. As QMS, platforms streamline the monitoring of compliance deviations and audits, ensuring trial processes align with regulatory requirements and standards. Question 5, why is batch and lot verification critical in certification evaluation? A, to create therapeutic dose response profiles. B, to confirm consistent drug quality and originality for regulatory approval. C, to identify biomarkers associated with patient safety outcomes. Or D, to detect placebo effects during clinical trials. So definitely the batch and log verification, why it is critical in certification, as the answer is B, to confirm consistent drug quality and originality for regulatory approval. As batch and lot verification ensures that each drug batch adheres to quality and regulatory standards, maintaining consistency and authenticity throughout the production process. And here we come to the end of this lecture. Thank you very much and goodbye.